biosciences and biotechnology, latest developments and opportunities. I invite Dr. Susan Epen and the club president onto the stage. Balagobal to formally introduce today's guest speaker. Introduction about this session's resource person, Dr. Susan Epen. Stanford University included Dr. Susan Epen in the world 2% top-ranking scientist list in the category Plant Biology and Botany in the year 2020, a decade after she retired from Bhabha Atomic Research Center, Mumbai in the year 2011 as a senior scientist and head of Plant Biotechnology and Secondary Products section. Currently, she is engaged in motivating scientific temper among faculty and students at Union Christian College, Alua, as an adjunct professor in Biosciences Group. She is a past president and member, Board of Trustees of Indian Women Scientists Association, and continuing her activities with them, organizing symposia, workshops, webinars, and other virtual activities remotely from Alua, where she has relocated in the year 2018. She is considered one of the pioneers in transgenic plant research, and her interest varies from plant biotechnology, production of secondary metabolites from in vitro cultures, and phytoremediation. She has published 140 research papers, contributed several book chapters, and presented papers in several national and international symposia. Her latest paper is on phytoextraction of uranium using vetiver plants in 2020. During an Indo-FRG bilateral program in 1986 at Free University, Berlin, she developed the first transgenic grain legume from protoplast using 3 different techniques. Later, she developed transgenic plants which could take up heavy metals and degrade organic pollutants. She was also involved in the Cotton Mini Mission project of Government of India and was a collaborator of several BRNS-funded programs all over India. Scientists from Russia, Cuba, Syria have come to her lab to get trained in latest techniques. She has been in the editorial board of international journals in vitro plant and BMC biotechnology. She received Elsevier Award in 2010 for the best quoted paper of the year, and the H index of her publications were in the 40s, which is considered outstanding. She is also a recipient of UNESCO and British Council fellowships. Red Dot Foundation, in partnership with the Office of the Principal Scientific Advisor to Government of India, listed her among the top 75 women in STEAM on 3rd March 2022 and felicitated her last year while we were celebrating 75 years of India's independence. She is featured in the 2nd edition of She Is 75 Women in STEAM, that is Science, Technology, Engineering, Arts and Mathematics. So with this brief introduction, I welcome our resource person, Dr. Susan Epen, to present. Thank you.
Thank you, Cynthia, for the kind words of introduction. It gives me great pleasure to be here to give a talk on biosciences and biotechnology recent developments and opportunities. I thank Mr. Vimal Venu and other organizers for inviting me to deliver this talk, and also Dr. Jayaraman for supporting me. Actually, I am not a career consultant in biotechnology, but since they have asked me to give a talk, I'm just giving a talk. I'll be speaking in English, and at the end of the talk, you can ask questions in Malayalam or in any other language. Now, the topic for today is biosciences and biotechnology, recent developments and opportunities. If you look at biosciences, there are so many different fields. You are familiar with some fields. I have included only some fields here, like botany, zoology, medical and health sciences, microbiology, biochemistry, biophysics, biotechnology, immunology, veterinary sciences, agriculture sciences, pharmaceutical sciences, molecular biology, cell biology, synthetic biology, genomics, proteomics, metabolomics, horticulture, plant breeding, bioinformatics, forensic sciences and criminology, genetics, virology, fisheries and ecology. So there are so many topics, but I have not included everything. So what is biotechnology? Biotechnology is a part of biosciences, and biotechnology has been practiced by your great-grandmothers and their great-grandmothers for many centuries, like making curd using lactobacillus, making wine using yeast, were practiced from time immemorial. This is actually biotechnology. But what is biotechnology? Biotechnology is the technology which uses biological systems, living organisms, etc. to create products. So biotechnology is an integration of several sciences, like even chemistry, physics, integration of natural sciences and engineering also, to develop a particular product, like an enzyme, or it can be cellulose or starch or any product using living organism. So biotechnology was known to your great-grandmothers and great-grandmothers, but that term was coined only in 1919 by Karoli Erecki for using biological organisms to develop a technology. So one can use bacteria, yeast, plants, animals, etc. to produce different products. Then there is genetic engineering, then cloning of plants and animals, fermentation technology, etc. to produce novel things. You can make drugs, biofuels, genetically modified organisms. You can make novel enzymes and innovative materials using biotechnology. Different fields of biotechnology are the following. One is the medical biotechnology. You all know how important it is to have a medical biotechnology, because when COVID struck us at the end of 2019, just because of the biotechnology scientists, the vaccines and diagnostics and RT-PCR, everything could be developed just because of medical biotechnology. Then there is animal biotechnology to improve the animals, the cow, sheep, etc. And you can produce hormones and other metabolites in animals. Then we have plant biotechnology, where you can improve our crop plants to produce more food under climate difficult conditions. Also there is environmental biotechnology. All of you are aware how the plastic burning in Brahmapuram in Ernakulam district had led to lot of poisonous gases in the air, and some of these fungi and plants can remove some of these dangerous components. During my talk, since you are not very familiar with all these topics, I'm just giving a general introduction and latest developments. And the second part of my talk, I will talk about the different opportunities, and then you can ask questions. Now, all of you have heard about the experiments of Gregor Mendel. Gregor Mendel, what did he do? In 19th century, he based on his experiments on garden peas, he said that there are characters. He was wondering how dogs give birth to puppies and cats give birth to kittens, not vice versa. Why it is like that? Why children look like their parents? So he initially did experiments in mice, but in monastery where he was staying, other people were not very happy using mice. And so he started working on peas. And he said, there are characters which are transferred from parents to offsprings. And he called these as factors. And one character will be dominant over the other. And these are known as the Mendel's laws of inheritance. You might have studied in your 9th and 10th standard. Now, some of the milestones in biotechnology. You know that cell division takes place in your body cells, in the plant cells by mitosis. What happens at the end of mitosis? A cell which is having 46 chromosomes, when it becomes 2 daughter cells, each cell will have 46 chromosomes, right? So this was discovered only in 1870. Then microorganisms, can you see it using the, without a microscope, can you see the microorganisms? No. So you have to use a microscope, and staining of microorganisms were developed by Robert Koch. Then Mendel's laws, after 35 years, it was rediscovered. Then chromosome pairing. Then T.H. Morgan did a lot of work on fruit fly. You would have all seen fruits. If you keep fruits for a long time, you will see tiny flies on the top of banana, ripened banana and all that. He worked on that and reached many conclusions about chromosome and genes. Then William Johansen coined the name gene. There is a beautiful experiment by Frederick Griffith on transforming principle. That is, a principle is moving. Suppose you kill a bacteria and some chemical is moving from dead bacteria to the living bacteria and cause infection. So then Alexander Fleming, all of you are familiar that during the Second World War he discovered penicillin, which saved the lives of thousands of soldiers. Then Beadle and Tatum, they worked on a fungus called Neurospora. Then animal cells were grown in vitro, plant cells were grown in vitro, and from plant cells you can get the complete plants. I have just shown the picture of a plant cell, an animal cell, a virus, and a bacteria. What's the difference between a plant cell and an animal cell? Plant cells have cell wall, whereas animal cells don't have cell wall. Plant cells have chloroplast, whereas animal cells don't have chloroplast. And bacteria, they are much, much smaller than plant and animal cells. And viruses, all of you are familiar with COVID virus. They don't, in environment, they are not living, but they have a genetic material. The moment they enter a human cell, the moment they enter a plant cell, they start becoming active, and they're using the host mechanism, they multiply and come out. So this is the chromosome structure. A chemical molecule called DNA is situated, arranged inside the chromosome very nicely, and genes are situated in the DNA. So this is a human chromosome. There are 22 pairs of autosomes and 1 pair of sex chromosomes. Watson and Crick, all of you are familiar with the DNA structure. DNA is a double helix, and in 1962 they got the Nobel Prize for that. There are 4 letters: adenine, thymine, guanine, and cytosine, and then there is a genetic code. This is a picture of Severo Ochoa and Arthur Kornberg, and they also got Nobel Prize. I happened to visit them. Now, 10 days back, one of the great scientists who discovered the restriction enzymes passed away. That is none other than Dr. Paul Berg. Because of Paul Berg, now we know that, we can do, all of you can work on molecular biology. He found out restriction enzymes, which are molecular scissors. Suppose you are given Fevicol and a thread and a scissors and two threads, one white thread and one red thread. You can cut the red thread with the scissors, and with the Fevicol, you can paste it into the white one, right? Similarly, because of the restriction enzymes discovered by Paul Berg and also the DNA ligase, you can cut the DNA at any point that you want and paste it with another enzyme called DNA ligase. This changed the whole programs in biosciences. In 1978 to 82, human insulin gene was cloned in bacteria. Do you know how insulin was produced up to that time? Diabetes 1 and 2 people, so many people need insulin. Up to then, from the slaughterhouse where cows and pigs were killed, their pancreatic cells were taken and insulin was isolated. But that is not sufficient, for what? For getting, because the diabetic people are increasing. So the gene from human beings was cloned into bacteria and they were grown and the insulin was commercially produced. So the insulin that you get in the market right now is produced by bacteria. Then you all are familiar with RT-PCR. What is RT-PCR? PCR is polymerase chain reaction. So Cary Muller in 1980, for the first time, he discovered polymerase chain reaction and he got Nobel Prize. Then you are familiar with the DNA fingerprinting. Suppose a crime is committed, they take blood samples, hair and everything, and they do DNA fingerprinting to find out who has committed the crime, isn't it? So very important in forensic sciences. Then genetically engineered plant in tobacco for the first time was developed in 1984-85. Then vaccines can be produced in a tobacco plant. So that was developed. Then tomato, if you keep outside, it will decay after some time. Some technology like antisense RNA was done so that tomatoes can be kept outside without decay for maybe 1 or 2 months. Then first gene therapy was established in 1990 and there was a human genome project, as a result of which the whole sequence of human genes in humans were sequenced and it is available for us to use it. Then cloning of Dolly. Initially it was thought that animals cannot be cloned, but for the first time a sheep was cloned in 1997. And embryonic stem cells can be grown in vitro. Then this is just a technique of gene cloning. This has revolutionized the whole biology. You can take a gene, you can see here, this pointer is not working too well. A red one, red color, you can see a gene of interest can be taken from another organism, like from a fungus or from a human being or from a plant, and introduced into a circular molecule called the plasmid and introduced into the bacteria or into the plant cell or into the animal cell, and the red portion will get integrated into the genome of the host organism. This is known as gene cloning. Now in 2010, J. Craig for the first time produced a small synthetic cell, bacterial cell. And in 2012, there are many techniques of genome editing or gene editing. 2012, 2 women, they did gene editing using a special technique called CRISPR-Cas9, which is a technique used by bacteria. So in 2020, the COVID-19 came, and how much of work biological scientists and chemists and other people have put in to produce vaccines, to develop diagnostics, etc. Now comes to medical biotechnology. So human health is always important for us, isn't it? Other than like animals, we are not bothered so much about animals because we want to cure the diseases and we want to prevent the diseases. As I said before, the whole human genome project was started and the complete sequence is available right now. It has got wide applications in oncology, in neurobiology to cure genetic diseases. The new vaccines were discovered, new antibodies, enzymes, etc. Genetically modified insulin, as I said before, for the first time was developed in 1978 by the company GeneTech. Then gene therapy, genetic testing. Suppose somebody is having a breast cancer, whether it is possible that the children, girls, daughters and granddaughters will inherit it, that is by gene testing. Then molecular diagnostics. In 2020, SARS-CoV-2 came and so many techniques were developed by biologists like diagnostics, prevention and new drugs for treatment. Then industrial biotechnology. There are so many applications of industrial biotechnology using cells, microorganisms. You can make enzymes. You can produce a lot of useful products in food, feed, detergents, textiles, biofuels, etc. Suppose you want to change the oil composition of coconut oil, you can do that. You can do metabolic engineering so that normally metabolites are produced in plant cells or bacteria and microorganisms by different steps. So each step can be manipulated by metabolic engineering. Then there is bioinformatics. It's a mixture of computer technology and biology to solve biological problems using computational methods. This it results in analysis of large amount of biological data. There are so many things like genomics, structural genomics, proteomics, etc., which can be used using computational biology or bioinformatics. You can even design new drugs using in pharmaceutical industry for treatment of diseases, new vaccines, etc. Then coming to plant biotechnology, you know how important it is to produce climate resilient crops to stand high temperature, drought, non-availability of water, salinity, etc. Many techniques are there for that, but the latest techniques is genetically modified plants and genome editing. You can see here the golden rice, which is having a beta-carotene gene, then a flavor-saver tomato, which can be kept on the shelf without getting spoiled for a very long time. You can modify your rice, you can modify your wheat, so many things you can do. But when you manipulate plant cells, it's very important to remove the cell wall. So the cell wall have to be removed, and that is called protoplast. You can bring about protoplast fusion. You can introduce novel genes into the plants and develop complete plant, and you can produce a transgenic plant. Then there are structural genomics, functional genomics, and the first plant whose sequence was completely sequenced is Arabidopsis thaliana, a cousin of Indian mustard, using sequencing techniques. Now this is developing transgenic plants using agrobacterium and using particle gun bombardment. You clone a gene and you see the machine there. You can bombard the gene into the plant cells and develop the complete plant. Using this technique, you can develop a BT cotton, flavor sour, tomato, etc. Then there is environmental biotechnology. As I said before, you can biodegrade pollutants like dioxin and anthracene, benzanthrene, etc. Using this technology. Biological organisms are used, especially some fungi and plants, for organic pollutants like anthracene, chloroform, carbon tetrachloride, etc., which are which many of these companies are throwing into the rivers and soil. Even toxic metals like cadmium can be contained. You can remove arsenic, which is a major problem in West Bengal. Then you can improve soil and solution. You can use microbes and plants to improve our environment. Then recent breakthroughs in biotechnology, I am talking about this so that you will have an idea. Unless you have a basic knowledge, you cannot decide which topic you should pursue. Then genome editing, I already spoke about the CRISPR-Cas9. There are novel techniques like base editing, prime editing. Then you can print an organ like 3D printed organs. Then tissue engineering, you can develop new vaccines. Then you can have personalized medicine. What medicine will suit you may not suit me. So according to your genetic constitution, you can develop new medicine for you and for me differently. Then targeted cancer therapy, then brain signals to audible speech, stem cell research, then artificial meat. Many people don't like killing chicken and cow and other animals. So you can produce meat in the test tube. You can produce egg whites, gene therapy products. Then all of you, maybe girls, may be especially interested in skin care products. You go to the beauty parlor. So many of these skin care products, instead of killing animals and isolating from plants, you can make in the test tube, like skin care products. Then of course, biopharmaceuticals. This is a bioprinting of organoids or organs. You can make a heart, a part of the ear, using laser-assisted extrusion technology, stereolithography, etc. in the laboratory. Then 3D printing can be used. It was initially used by Charles Hull in 1980, but in late 1990s, it came into healthcare to make dental implants, custom prosthetics, and kidney bladders, and it is an important technique. Applications of 3D printing, there's a lot of requirement for organs, organ transplant, like kidney, like liver, like heart. Then for drug delivery, it can be used for studying the mechanism. I'm not going into the details. Then vaccines, there are different types of vaccine. I'm speaking only about mRNA vaccine. One lady called Catherine Carrico, who was a pioneer in mRNA vaccine. She developed this vaccine so that it is very useful. In the US, it is used for as a vaccine, mRNA, not only for COVID-19, for several other diseases. This lady has developed the vaccine, and one day she might get a Nobel Prize. Then meat can be cultured in vitro. You can see the picture. You take stem cells from a cow and grow it in the petri dish and make it into myotubules, then muscle tissue, then you can make a hamburger. Okay, it will be available in a KFC or in other stalls. Then lab-grown meat has got wide applications. Meat grown in lab, it has a very good environmental impact. It is good for animals, and it will mitigate climate change. Okay, now I'm not going into the details, but this Israel started a farm and fork restaurant called Chicken, and in December, Singapore restaurant started selling hamburgers. Pork, beef, chicken, lamb, bison, etc. can be made, and hamburgers can be made using this technique. In India also there is a startup. So biopharmaceutical, if you are good in chemistry, you can combine the knowledge of chemistry and modern biotechnology to produce novel products. You all are take aspirin sometimes, at least when you get fever, right? It is from a plant, but long time back in 1893, this was synthesized by Dr. Bayer. So pharmaceutical are directly isolated from plants, like for example Taxol and Camptothecin. They are isolated from plants, and these plants will get extinct one day because these plants are cut off by the companies, and you can make all these products in the test tube. So many protein-based pharmaceutical can be produced like insulin, enzymes, vaccine, antisense, oligonucleotides, antibodies, peptide, etc. can be produced by biotechnology. Then recombinant DNA can be used, for making so many products like skin products, cosmetics, hair care products. You don't have to kill a shark to get hyaluronic acids, squalene, collagen, etc. You can produce them in a bioreactor using DNA techniques. Then genome editing, I was telling there are many techniques for genome editing, but two women scientists, Jennifer Doudna and Emmanuel Charpentier, they published in 2012 and they were awarded Nobel Prize in 2020. It generated a lot of excitement in biology, and people are working in the medical field, in the plant field to develop new plants and to cure genetic diseases. These are the two ladies who got Nobel Prize. Then I'm not going to the technique. So researchers use this technology found in bacteria to edit your DNA. The beauty of this technique is that DNA, one single nucleotide, can be altered using this CRISPR-Cas9 technique. Because some genetic diseases like sickle cell anemia, hemophilia, etc. are caused by single nucleotide mistake. It has changed. So that can be changed using CRISPR-Cas9. This is a technique. And I am just talking about sickle cell anemia. What happens in the sickle cell? You can see the blood cells, red blood cells in a sickle shape. That is due to a small mistake in the DNA. So that is corrected because using CRISPR-Cas9, the gene is, the DNA is inserted into the particular cell and put it back into the bone marrow, and the cells that are produced after transplantation of the modified cells using CRISPR-Cas9, they are healthy red blood cells which are normal. So this technique was used to first lady to be cured is Victoria Gray, and she was cured of this sickle cell disease using CRISPR-Cas9. Then other diseases also are being cured. Metabolic diseases, neurodegenerative diseases, hereditary eye diseases, cardiovascular diseases which are due to a single gene mutation can be cured, but everything is under a very primitive stage right now. Then now coming to the career you want to choose. You can, I don't know what you want to become. You can become a research scientist. I don't know how many of you want to become a research scientist. You want to become an environmental consultant? Do you want to become a pathologist, data analyst? You want to develop a new drug, agriculture biotechnologies? Or you want to become a college lecturer, then a professor and principal? Or you want to have an entrepreneurship? You want to start a new business by starting a new startup? All these things are possible, okay? So you are at the crossroads. You can see in the second picture, you are standing there and there are 4 roads to take. Which one you will take? You don't know where to go, okay? But without your involvement, you cannot succeed. With your involvement, you cannot fail. So you are at the crossroads, and it is for you to select the correct road to reach your destination. There are so many universities. If you look at the different universities, 436 universities offering biological courses, deemed to be universities 126, central universities 54, private universities 388. They have 860 microbiology institutes, 384 biotechnology institutes, biochemistry institute, when biology taught in different colleges like this. Then now you are going to take one of the roads. Which road you will take if you are taking biology? You want to do a science-based oriented program, or you want to take a technology oriented program? If you want to take a science-oriented program, you can do BSc, that is undergraduate, and MSc, that is postgraduate, do PhD and join a job. If you want to do industrial-based technology, bioscience, you can take industry-based engineering courses like BTech, BE, then MTech, ME, and PhD and job. And where do you want to do it? You know, as a student, you have to select where you want to study BTech or MTech or BSc and MSc and PhD, where you should do. The best institute to do is Indian Institute of Science, Bangalore. That is a top NIRF ranked institute in India. I'm not talking about the foreign institutes because somebody is talking after me. Then there is ISER, Indian Institute of Science, Education and Research. They are very good institutes for doing scientific work, BSc and MSc and MTech and BTech and all that. Then, of course, in our time, IITs were having only engineering science. Now, IITs have started, you know, they also have started biological sciences, biomedical engineering, biochemical engineering, biotechnology, etc., etc. Then there is University of Mumbai and the Center for Excellence in Basic Sciences, sponsored by Department of Atomic Energy. There is a good course, BTech and MTech combined, or MSc and BSc combined, and its sister institute called NISER in Bhubaneswar. Then you have choice of taking private colleges, government colleges, etc. There are 8 institutes. If you look at ISER, Indian Institute of Science, Education and Research, there are 8 institutes: Pune, Calcutta, Mohali, Bhopal, Thiruvananthapuram, Tirupati and Berhampur. I have visited Pune, and that is the best institute for ISER for doing BSc, MSc and PhD, other than Indian Institute of Science, Bangalore. They also have BS and MS and integrated. Sometimes they are combined. A 5-year course with a BTech and MTech, 5-year course with BSc and MSc, and then PhD. Subjects include microbiology, advanced genetics, genomics, etc. Okay? Structural biology, physiology, immunology, etc. Then Bangalore, Indian Institute of Science has got a BSc, MSc courses and PhD in basic biology, neurobiology, ecology, biochemistry, etc. It has got the highest ranking in India. Then the University of Mumbai, CBS, Department of Atomic Energy, it has got a sister organization called NISER in Bhubaneswar. There's a common exam, integrated MSc program is there, and there's a common exam also. You can write the exam and get into that, and you get, I think, stipend also, good stipend. I was teaching in this course maybe for 2-3 years. Then IITs, 23 IITs are there currently, and IIT Madras is supposed to be the best, but I don't know whether it's very good for biotechnology. I'm not very sure. Then IIT Delhi, IIT Kharagpur is very good for biology. IIT Kanpur is good. Gauhati IIT is there. Hyderabad, Indore, Varanasi, Palakkad, etc. I don't know about Palakkad whether they have biotechnology, but IIT Madras has BS and MS in biotechnology. Then BS, BTech, MTech in biological engineering, which will have 5 years course. Then bioprocess engineering. IIT Madras is having several courses. Then IIT Kharagpur is having BTech in biotechnology, MTech in bioprocess engineering, PhD in biotechnology. IIT Mumbai is specialized in biomedical engineering. So all these opportunities are open for you. Then IIT Delhi has BTech in biochemical engineering and biotechnology, BTech and MTech dual degree in biochemical engineering and biotechnology, and PhD in biochemical engineering and biotechnology. IIT Kanpur is very good. BTech in biological sciences and bioengineering, which is a 4-year course. BTech plus MTech, MTech itself, biological sciences and bioengineering, and also PhD. Now coming to Jawaharlal Nehru University in Delhi, this is one of the best institutes. It has BSc, MSc and PhD in life sciences. Then all of you might have heard or might not have heard about Institute of Chemical Technology in Mumbai. It is a very good institute for biotechnology and for food sciences. They have a beta course, MTech course, etc. And private colleges, there are several private colleges. I cannot even put them in the list. You can do their BSc, MSc and PhD. Then Amrita School of Biotechnology, Vellore Institute of Technology, Amity University, SRM University of Science and Technology, Manipal Institute of Higher Education, Birla Institute, BITS Pilani, Goa and Ranchi. There are so many institutes. And after this, you can do PhD. Okay? So this is a very complicated thing. You are doing life sciences. You look at the top, you can go for BSc or MSc in genetics, or BSc or MSc in forensic sciences, BTech, BSc, MTech in industrial biotechnology, MTech in biomedical engineering, BPharm or MPharm. Then you can do BSc in botany or zoology, you can do BSc, BTech in environmental engineering, you can do biochemistry, biophysics, immunology, microbiology, agriculture sciences. You can choose anything that you want. So bioscience technology is a very complicated technology. There are people who take biotechnology even after medicine. My friend, Dr. What is her name? She was even Dr. Priya Abraham, she is a virologist who was a head of National Institute of Virology. She is a medical doctor. Even that other lady from Reliance, she is also a medical doctor, but working on stem cells. So medical biotechnology, you can work even if you are a doctor. Pharmaceuticals, drug delivery devices, you can combine with biomedical engineering, diagnostic devices, medical biosensors, then nutraceuticals, veterinary technologies, biomaterials, bioindustrial technology, biosecurity technologies, then agrobiotechnology, fermentation, industrial bioprocessing, food processing, pharmacologically active, genetically modified organisms, agribiotech. The thing is very, very vast. So you are at the crossroad. Now, how will you move ahead? You have so much of information. Now it is so difficult for you to choose. In our time, we had no problem because no information was available. So your problem is your parents want you to take a particular course, but you don't want to take. And your friends are going for engineering or BBA or commerce. So peer pressure is there and their influence is there. Parental pressure. Too much information is available in the net. And you don't know what are the strengths and what are the weaknesses. And you worry about failure. Suppose you don't like a subject, and if you take the subject, what will happen? Then some of the courses are very costly, like private institutes especially. Can your parents support you? Then what are the attributes? You should look outside your textbooks. You should be open to learn. You should be well informed. You should be confident and optimistic. Decide what you want to do because it is your life. It is not your parents' life. What parents could not achieve in their lifetime, they want their children to achieve. That is not correct. So set and define your goals. Proactive. And there may be students who have already studied, completed this course. Discuss with them. Now, real scenario is you can have academic research and PhD, or you can have an entrepreneurship. So you need specialized skills for doing research. Like you should be good for bench work. You have to work for several hours together. You have to do literature survey. You have to do scientific writing. Then there is no barrier in science. You have to know physics, chemistry, even engineering. It's an interdisciplinary learning. It's not just biology. You should have general skill like work planning, execution, troubleshooting if it comes. Then you should learn to work in a team. You should have a work culture. And you should have professional knowledge, competency, honesty and integrity, and accountability. These are the foundations if you want to do academic research. Suppose you want to do entrepreneurship, you have freedom to make what you want to do. Freedom to do research on what you want to make finally. So you will get satisfaction through financial and societal benefit. You should have managerial skill if you want to start a small startup, isn't it? You should be able to market You should be able to do selling. You should develop and improvise. So you can go into industry positions or teaching positions or research positions if you want to take up a job, or you can do other things in startup. So real scenario, the opportunities and what are the benefits to society. Then in academic research, I was in academic research, so significant research can be done and information can be given to public. And whereas in entrepreneurship, you will have the product, you can create your skills to develop products, you can have create employment opportunities, jobs for other people, okay, support scientists, then build ecosystems, support knowledge and technology transfer. These are some of the things. So academic research, PhD, I don't want to go into the details. Actually, these 3-4 slides are meant for postdoctoral PhD students. There are several institutes for advanced learning. All India Institute of Medical Sciences in India, Department of Biotechnology Institutes, 15 institutes are there. Count CSIR institutes, 16 are there. Department of Science and Technology, 11 institutes. Indian Council of Agriculture Research Institutes, 4 are there. 4 deemed universities, 65 institutes, 14 national research centers, 6 national bureaus, 13 project directorate, then IITs, many are there. So BITSA is there, then TIFR is there, Tata Memorial Center, Mumbai, Delhi University, campus. So many universities are there. It is for you to choose. So this is not actually meant for you. This is for BSc and MSc students. There are so many exams to pass if you want to join a PhD and all that. So I would like to tell that it is for you all are wanting to join somewhere, and you may be having some ideas in your mind. So please go to the internet and search what are the courses available. I will leave the slide here with the organizers. If you want, you can copy the slide, but you can decide what you want to do. You can go to the internet and search which all courses are available in which all universities, how they are rated, and then you can choose what you want to do. Then for entrepreneurship, there are many certificate courses, diploma in industrial biotechnology, certificate courses in management, then plant tissue culture, microbial quality control, fermentation technology, short courses, 2 months, 3 months in some of these institutes, fermentation technology, bioinformatics, pharmacovigilance, clinical research and data management, human and animal ethics, intellectual property, patenting. One can go for patenting also. IIT Kharagpur has a good patent course for students who have completed their graduation. So there are, these are some of the important links. Before I conclude, I would like to tell, you can ask questions. I would like to tell that, the current education system is such that, when you come out of your 12th standard or BSc or MSc or PhD, you are like a machine-made product, industrial product. You cannot think before outside. You have to be innovative. You have to think outside what is being taught. You have to read outside. You have to go to the library and use that opportunity to develop your skills. You have to be innovating. 21st century need innovation and new skills. Some of the children where I was staying, like in Anishakti Nagar, they have reached very high heights, like Parag Agarwal. He was my colleague's, my classmate, batchmate's son. He was the CEO of Twitter. So all of you can reach any heights. It's not a big thing. So you should have the motivation and you should plan accordingly. Okay? And I can leave the slides here if you want. It's not actually meant, I don't know whether it's meant for you, but if you want, I can leave it. It is anyway here. You can copy it or do whatever you want. Okay? And all the best. If you have any questions, I can answer. Thank you.
Thank you, ma'am. If any of the children have any queries, you are welcome. Anybody wants to ask anything to ma'am? Shall I pass the mic?
Even after medicine, one can go for one or some of these courses you can attend and shift your field to research.
For the specific colleges for biotechnology, are there any scholarships that we can write so that we can
In the University of Mumbai, CBS course supported by Department of Atomic Energy, there are scholarships. All are given some stipend enough to live and take care of everything. Right now I don't know because I was teaching there some 2008 to 2010 or something like that. So it may be still there. You go to the site and find out. Even on that nicer Bhubaneswar also will be giving stipend. Good enough to live, like I have food and everything like that. And then directly they take you for PhD also.
Thank you, ma'am.
I think all of you, there is a lot of information available in the net as well as everywhere, and you are all confused. Your parents want you to, I don't know what they want you to do. You can go for BTech or MTech or BTech or biomedical engineering or BSc or MSc, whatever you want. It is for you to come up. Whatever interest you have, you should develop that. Even if it is paleontology or ecology or anything, you should be the best person in that field. You should be, even if it is the most useless subject, what others think, you should do your best if you like it, and you should take a subject which you like. I remember one of our neighbor's son, he had come to my house. I had just taken a tuition for not tuition actually, along with my son. He was in 10th standard, he was not understanding much of biology, 10th or 12th I think, biology, and 1 hour I had taken on cell biology, division of chromosome, so chromosome division, mitosis and DNA and all that. And then his father was an electronic engineer in BRC only. He went for, I think 2 years he studied in a BS college for engineering. Then he realized he should not continue that. And then he left it and went and joined for medicine. Then after medicine, he went abroad and he went to research, and now he is doing very well in some medical field, but research. Then somebody asked, why did you change after 2 years? He told his parents, Susan auntie had taught me for 1 hour when I was in 12th, and that's why I liked biology and I changed. So your teachers really, influence you. So it's very important to teachers to have good teachers. They will motivate you, and you are like a small diamonds which are not polished, but the teachers will polish you, and they will motivate you. So you should have a motivation, you should have an ambition, and what is today, what is applicable today may change tomorrow. No? Today there's a lot of demand for one particular company, one particular field, but tomorrow it may change because of artificial intelligence and other things. So cannot guarantee what you take, but you should take what you like. Now people are changing jobs every 2 years or 1 year or anything, something like that. Now we have all worked, Dr. Jayaraman and myself, we have worked in the field for nearly 40 years without getting bored, and we are happy. So that satisfaction should be there, happiness should be there. It's not just the money, it is a satisfaction. Okay? If there are no questions, should I wind up?
Anybody else? That side? Any doubts about what ma'am presented, or any other, beyond that, if there are any questions?
If you have a basic understanding, basic knowledge of biology or biology especially, you can build it up, because just knowing advanced technology is not enough. You should have the basic knowledge. If there is a foundation, then only you can build on the foundation, right? So you should have the basic foundation. So better to do basic courses like microbiology, botany, zoology, biochemistry, biophysics, etc., and then go into advanced fields.
If there are no other questions, shall we wind up the session? Okay, ma'am, thank you so much for a very informative
Thank
session.
you so much.
On behalf of Lions Club also, I thank you.
The Lions Club of PALGHAT recognized the presence of Dr. A.P. Jayaraman. We had a wonderful session of this nuclear scientist yesterday. Thank you for visiting us today also. Thank you, sir. We'll be moving on to the next session led by Manu Rajagopal on the topic Overseas Education Myths and Realities. We'll be starting it.